due to release of enzymes from lysosome into the cytoplasm. Cell death is due to release of enzymes from lysosomes into the cytoplasm. So these enzymes start to digest the other organelles, cell membranes, and then finally cell is dying. Okay, if you have no questions, we can continue. Right, this is another question, MCQ questions related to endoplasmic reticulum and lysosome. So, function of lysosome and endoplasmic reticulum are stated respectively. So you need to find what is the function of lysosome out of this ABCD, and what is the function of endoplasmic reticulum out of this ABCD. Okay, shall we try to find the answer for this question? First you try. Okay, correctly identified. Yes, if you look, the production of transport vesicles, we have seen endoplasmic reticulum. Its function, one function is production of transport vesicles. Now, if you spend the time to remember all the functions given in the notes or resource book, you have the set of functions everywhere. Now I had to bring all this information to answer this question. And we know this production of cell wall components such as cellulose and pectin, this is done by the Golgi. It's a function of Golgi. It's not a function of lysosome or endoplasmic reticulum. Removal of residual material from cells by, so we exactly same wording we learn. Residual material. What are residual material? There are some leftovers after digestion. They can release out of the cells by exocytosis. So lysosome help to do this. And this one is the function of lysosome. And sometimes, you know, just for the easy remembering purposes, sometimes we try to say lysosome, Lysol. Lysol is the cleaning material you can find in the supermarket. So same way the lysosome is the cleaning guy in the cell. It just clean everything and either recycle or release out. Lysol, lysosome. Production of transport vesicles. This is function of Golgi, SCR, and RER. So RER has this function as well. So then respectively mean which one is the first. Lysosome function is the first, D, and the second one is the endoplasmic reticulum, B. That's why it's used this word respectively, D and B. So D and B is here. So this is again example of practice. To practice, You need, number one, remember the content. When you remember the content, first maybe you might be wrong because when you write, when you make the answer wrong, but more and more practice, at one point, the chance of getting wrong answer is getting zero. So that's a way you have to really use these things, right? Okay, done. Okay, I will also discuss with you example of a structured question, because always wherever we have questions, I put it here and we discuss the question. One thing is how you practice your knowledge to find the better answer. Now, look at this structure. It is a drawing. It's not an electron micrograph. It is not the real picture. It's a drawing. So in this drawing, they have actually some arrows as well. You can see this arrow is not very clearly, not very much clear, but you can see this one. So I draw it again. This is called vesicles, right? These are called vesicles. Vesicles are generally membrane-bound, small round-shaped structures, which might have many things, enzymes, proteins, whatever. They're called vesicles. Now this vesicle is moving with this arrow to this side. And here there is again vesicles. They are going out. Right. This organelle, if you go to this drawing here, the first one I did here, it just representing this one, the Golgi. An electron micrograph, it is like this. Then you can write this structure is called organelle. You see how in the same exam, same paper, same place, then again subcellular structure. They change the word again, but the words are the same. Organelles and subcellular structure, this is same. The two words are same. What is the organelle? It's a Golgi. You can write Golgi bodies, Golgi apparatus, Golgi. It just accepted everything. What is the subcellular structure which produces new vesicles that join with this organelle? And now, if you look at the resource book, that much detail is not there. The protein transport process is not very clearly highlighted there. But I explain you today. I explain you today this whole process. This one. I said the nucleus is this side. And then the first thing is the ribosome. Function is synthesize protein. Number two, the protein is moved to the RER now. RER, we can say it's modified, but it's pack and make new vesicles. Here you can see this one is the vesicles. Now the vesicles are moving to the Golgi. It fuses with Golgi. And after that, these vesicles made by the Golgi, like here, they're going out. They're going out as bulk, like in fact with many, not one single, it's not a single molecule, maybe 1,000 protein molecules going out. This is the protein transport process. So here the question, it asks actually that one. What is the subcellular structure which produce new vesicles that join with this organelle? Now we can, if the RER is this side, let's say this is RER, and that's a ribosome, and RER can pack the proteins and make nerve vesicles, and the vesicle is moving to this side. That's RER. So the subcellular organelle is a rough endoplasmic reticulum. Now state two functions of the organelle shown in the above diagram. It's asked, actually now you have to identify Golgi. If you correctly identify, we have three functions, right? Golgi, pack, transport, distribute the material. It also synthesizing cellulose and pectin. It also synthesizes lysosome. There are three functions. So out of these three, you can write these three functions, right? Let's say if I write a function like this, it modifies the protein. This is perfectly correct. It modifies the protein perfectly correct. But I would like to say you can, let's say you can write it's like this into the glycosylation. You can write the glycosylation of the protein. Let's say you write glycosylation of protein. It's correct, perfectly correct. But you know, we adhere to our guidelines. In the guidelines, Golgi has three functions: collect, pack, and distribute of material, manufacturing cellulose and pectin-like molecules, produce lysosome. So you have to write only that two functions. Out of these three, you just write the three functions because we have some kind of rules. I mean, you can have a three textbook, you can get so much of advanced knowledge, you write it here, you risk your marks, right? So keep in mind, the two functions are listed there, 1, 2, 3. So out of that, you can write pack, transport, and distribute the material. Number one, manufacture products such as cellulose and pectin. Number three, produce lysosome. So out of these three, you just write two. Okay. There is another question. Shall we try this? Okay, again, focus. Incorrect. Regarding smooth endoplasmic reticulum. Okay, if you little bit of, we go again, check SCR. Right, the SCR looks like this. an electron micrograph, again, they don't have very clear structure. It's like a kind of a network of some kind of tubules like things. It synthesizes lipid, including steroid and phospholipid. So one function is it produces steroid hormones and some lipid and phospholipid also. Metabolism of carbohydrate, that means some carbohydrate breaking and making, synthesizing, anabolic and catabolic reaction take place. Produce transport vesicles within the cell. So it's not going out like lysosome or Golgi. It produced a vesicle within the cell, involved in detoxification, store calcium ions. And yes, the 5 functions. Store calcium ions, involved in detoxification, produce transport vesicle within the cells, metabolism of carbohydrate, and production of phospholipid and steroid-like things. So these are the 5 functions of SCR. Okay, if we look now the 5 functions of SCR, store calcium, yes, because muscle contraction, it's important. Most of the muscle cell SCR, they store calcium. And anything toxic that we ingest, we take from our mouth to the body, if that one is toxic and is going to the cells, then the SCR, they involve in neutralizing it. Detoxify mean the toxic compound, make it non-toxic. And when we look at the structure, I showed you the video as well. It's like a RER like this. They make the network, and at the end, it make again network of like tubes, cylinders. So it's a network of tubular sacs. So tubular sacs, correct. And we have seen it synthesize lipid, oils, phospholipid, and hormones. So phospholipid is a lipid combined with phosphate. This is lipid with carbohydrate of cell membrane. And we have seen again they are doing this function, and we haven't seen presence of membrane-bound enzyme. We haven't learned anything like this, presence of membrane-bound enzymes, because it's incorrect. So we have a confidence that we have all these CR5 functions, and 2, 3, 4, 5 are the function of SCR. Then you can go for what is not sure for you. You haven't learned such thing in SCR, so you can go for this answer. Now again, I'm telling you, you need to strongly remember by numbers the function of this organelle. This has 3 functions, this has 5 functions, this has 4 functions. These are the things. That's the effort from your side. The guidance, everything is given. Your task is to remember this function of organelles. Okay, again, we are coming to these microbodies. So microbodies, micro mean tiny, micrometers, micrometers, bodies are some structures. So we cannot distinguish them from the lysosome-like structures. Again, some kind of a structure like this. So they are peroxisomes, glycosomes. That's why I did not precisely include any micrograph, electron microscope picture because they don't have very specific structure. Okay, then what are the functions of Glycosome, Peroxisome and Glycosome? First thing is, what is the difference between these two? Are they same or are they different structures? A little bit of slightly confusion is here, but we avoid that confusion. We can say glyoxysomes, they are in the plants. So glyoxysomes can be found in plants. What is exact function of glyoxysome? In plants, they can convert lipid into carbohydrate. Convert lipid into carbohydrate in plants. The function is convert lipid into carbohydrate. So let's say sugars, carbohydrate sugars, they can use as energy then. Then the peroxisomes. So peroxisomes, they are doing important functions. Detoxification of hydrogen peroxide. Okay, what is hydrogen peroxide? H2O2 is the formula. H2O2. You can buy actually hydrogen peroxide from pharmacies because they are used for some diluted hydrogen peroxide is used for the wound cleaning and some other purpose. So this is the compound, right? Now question is, if you are interested, if they are toxic, so let's say if this is toxic to the body, why body produce hydrogen peroxide? So peroxisome can detoxify it. I think I will not spend too much time because it's a little bit going unnecessary information, but there is one thing. You will see this in photosynthesis, when you learn in photosynthesis. There is a word, this word is written in the notes, reactive reactive oxygen molecules. Now, what is a reactive oxygen molecules? Having a space to write here. So I use the space here, this side. You know, oxygen, if you take oxygen atom, this oxygen atom has 8 electrons, right? 2 here and 6 around it. 1, 2, 3, 4, 5, 6. There's a good example is given in plants, because plant is very specialized to absorb the light for photosynthesis. So when light is absorbed, in case this oxygen atom, especially the UV light, there's a chance of losing 1 electron. This electron is going out. 1 electron is going out. If this electron is going out, this oxygen has this kind of the negative which is lost. It lost the negativity, and it's right like this. The oxygen atom is molecule is right like this then. So it lost 1 electrons, and they are very reactive. This molecule is very reactive. It's dangerous. It can damage cells. It can damage other molecules. So to remove these molecules, this molecule convert into hydrogen peroxide, and after that hydrogen peroxide convert into again oxygen and water. I will explain again, don't worry this one. That's what I want to explain here, that hydrogen peroxide. Why it is detoxify hydrogen peroxide, why hydrogen peroxide is formed in the cells. Glycosome, again, fatty acid to carbohydrate, right? Okay, that's it. Another one I just want to tell, but it's too early. Another function of glycosome is they do something called photorespiration. Let's say the word, first I write it. Now, difficult thing is we can't explain photorespiration at this point. Reason is it is the mechanism we're going to learn in photosynthesis. So I think you have to tolerate a little bit. You remember this word photorespiration because it's not a single thing to explain. It's a pathway. Pathway means one thing formed here, it goes to another place, and goes to another place like this. So the photosynthesis takes place in the chloroplast. Then one molecule is going to the peroxisome. Then that molecule is going to the mitochondria. Then after that, that molecule return back to the chloroplast. So this whole story is written as photorespiration. So glycosome, mitochondria, chloroplast, 3 organelles important for photorespiration. Okay, let's see answer for this. Glycosomes transport of residue material out of the cells. No, glycosome convert fat to carbohydrate. Smooth endoplasmic reticulum, production of transport vesicles. We have said several times this one. Yes. SCR, RER, Golgi, they produce transport vesicles. Now, rough endoplasmic reticulum, metabolism of carbohydrate. So, metabolism of carbohydrate, we have again learned like a production of glycoproteins. Right? Okay, this one, nucleus, synthesis of glycoproteins. No, glycoproteins, that's wrong. Smooth endoplasmic reticulum, the production of transport vesicles is correct. Rough endoplasmic reticulum, metabolism of carbohydrate, it's actually the metabolism part is not there. It is only the production of glycoproteins. So we can't say it's a complete metabolic event, right? It's not a complete metabolic event. It's not. And this is wrong. Nucleus, look at 5 functions. They have not synthesis glycoprotein. Glycoprotein synthesis is part of rough endoplasmic reticulum. The peroxisome, they do a photorespiration. Now I did, okay. Yeah, I think I got this answer. This one is a 2021, the previous exam. There is a small mistake of this question. 2 is correct, 5 is correct. Mistake can happen. I mean, even exam, any kind of like A-level like exams, there are some mistakes is happening. So they have a 2 choices. Good for you. At least chances are high that you can get the answer. If you answer 2 or 5, it's correct. So it's a correct combination, right? Okay, next. So we look this should work. Yes. You can see a cell and you can see a shape of the mitochondria. So what I want actually, why I want to show this video, the mitochondria is, if you look, it's just moving, just like a bacteria, see? It's just like a moving. And the mitochondria has the special shape like this, like a cigar shape. And when you see this mitochondria, you might, if I don't show this word, you can get, is this bacteria, is this animal, kind of a small animal, is a small worm, like this impression is coming. So, and you can see a thousand of small red color things are going out. These are ATP, they are called ATP. Adenosine triphosphate. We learn adenosine triphosphate in biological molecules. They are the energy molecules. So it just produce energy and release these ATP molecules. So inside is like this. They have 2 membranes, outer membrane and inner membrane. As I said, nucleus, mitochondria, chloroplast. They have 3 membranes. Double membrane. I'm sorry, double membranes. And you can see inner membrane make some extension into the space here to increase the surface area. That means inside membrane, surface area increased by making this folding. These foldings are called cristae. And there's a large empty space that is called matrix. Okay, the outer membrane and the inner membrane. Now you can clearly see inner membrane folding and the matrix. So space is called intermembrane space. Outer membrane and inner membrane space is called intermembrane space. So inner membrane, there are proteins located. This is important for the next coming up lessons, respiration. So there are proteins located in the inner membranes. Right? Okay, that's it. It's very important to see mitochondria under the electron microscope. So it has this outer membrane and then the inner membrane. The inner membrane make just lost quickly. Inner membrane make the foldings like this. And you can also see in the video, there are some proteins attached to the inner membrane. And this folding is called cristae. And the inside is called matrix. This is the outer membrane. I just write M membrane, inner membrane M. and the space is intermembrane space. Okay, there is two important things. One is 70th ribosome, other one is circular DNA. You need to remember all these components, and mitochondria is extremely important. So take a piece of paper, and draw the structure and label it until you remember it well. So I allow you to quickly copy this one because we have enough time. So we have, if you maintain a nice a notebook, this notebook will be different to certain other notebook because you have a drawings and important things that will be a short note. So you practice that one many times, and then you can have the resource book. Then you can go through it again. It's less text, but you have all this information. Now, important question is, when I raise, it looks like a small organisms like worm or bacteria. And why they have a 70th ribosome? And why they have a circular DNA? So that's something the curious, because the animal cells, eukaryotic cells, plant cells, they have a 80S ribosome, but mitochondria has 70th ribosome. We'll find answer for this one in the next session. Right, I will again write the function of mitochondria, and also we find answer why mitochondria has a 70th ribosome and circular DNA. The function